so here we go. DNA structure and replication. Now, we put these together because I don't know if you picked up on this in the movie about Watson and Crick, but I mentioned this a few times also. Watson and Crick did not discover DNA, okay? They did not even discover that it was the genetic material. That was Griffith, Avery, and the Hershey-Chase experiment, okay? What they did discover was the structure. So Rosalind Franklin and actually Maurice Wilkins, we should say, so because he actually was in on this too. And I doubt anybody said he was the major contribution, but he was there and he did make a contribution to it. Maurice Wilkins, and then of course Watson and Crick. What exactly did they discover? They discovered the structure of DNA and the way that it replicated. Okay, so now this is important. I don't know if you saw them, and I mentioned this yesterday. They showed Francis Crick doing this kind of thing when he was showing off the model. This has to do with the replication of the way that the DNA, what the key was here, the structure suggested a method for DNA to copy itself. And that was the key. Because the genes, your genes, in order to get passed on to your children, have to be copied. And this is interphase in mitosis. Well, that's when they get passed on to other cells. We're gonna learn about how they get passed on to your offspring in the next unit. But for now, we're gonna focus on structure and replication. So structure, all right? Here's the basic parts of DNA. You have the phosphate group. I'm just gonna go briefly through this. In this next assignment, 24, you're gonna look at this in depth. There's a phosphate group, and the phosphate group is bonded to a sugar, okay? And the sugar has a pentagonal shape, so that's why I drew it like that. It's got carbons, it's got an oxygen in there, it's got some hydrogens. You got the phosphate, you got a sugar. Now there's another phosphate, so this is the phosphate, what's called the phosphate backbone, and then this will be bonded to another sugar, and so on, okay? And then it goes down like this. And by the way, what I'm drawing is in your book on page 175. This is the basic structure of DNA. And you are going to have to know it for the test. And you're going to be dealing with it on this assignment, also assignment 24. All right? So there's a basic, this is what's called the backbone. And there was one key thing that Rosalind discovered in her research, and that was that the phosphates were on the outside of the backbone, and that was a key part of the structure so that they could figure out how to build the whole thing. Now, attached to each sugar is what we call a nitrogenous base. Now, I would not hold you to these shapes. So, like, for example, if I asked you on a test to draw this, I would not expect you to be able to reproduce all these polygons exactly, okay? But I would expect you to know what is bonded to what. Follow me? So like that the nitrogenous base, this is called a nitrogenous. That just means it has nitrogen in it. Base, what do you think that means? It's not an acid, right? It's the opposite of an acid. So nitrogenous base, it's basic. If you put this stuff in water, right, this compound, this nitrogenous base, it would have a basic pH higher than 7. Now, there's different kinds of nitrogenous bases. The first is called adenine, and we use the symbol A for that. And then the second kind of base has almost exactly the same shape. It's only slightly different, and it looks like this. Notice, though, I do have, like I said, I'm not going to hold you to memorize the shapes of the polygons, but it's a hexagon and a pentagon. You can see that, right? And then the next one is different. The next one is just a hexagon. Whoops. And then another one. Yep. All right, so the second nitrogenous base. Now, all of these are nitrogenous bases, okay? 4 different ones. Adenine, the next one is called guanine. And that has a symbol G. And then the next one is called cytosine. And you are going to need to know these names. And then thymine, which is T. AGCT or ATCG. And there was this whole thing in the movie about the A with the T and the C with the G, right? And they were at a dinner when that guy was insulting them and stuff. That's what they were talking about, is they're talking about how much of these compounds, of these molecules, were in the DNA. Now here's where the replication comes in. So this is the basic structure of 1/2 of the DNA, all right? But it turns out that there is also another side, that what Watson and Crick figured out is that adenine matches up with thymine and guanine with cytosine. So let me draw another version of this. A little bit, I'm just going to make it a little bit smaller, so I'm not going to show the shapes. Okay? If this is adenine, guanine, cytosine, and thymine. See, that's a simplified version right there. You follow me? Simplified version, I don't have all the same shapes. It turns out that adenine attracts thymine, and guanine attracts cytosine. So what happens is that we get another strand matching up with this one. Okay, and this goes on and on and on. And these are joined by hydrogen bonds. Hydrogen bonds are like what occurs, that's what holds water molecules together. Now these aren't water molecules, but it's the same force. So what you have is here, look at this. You actually have 2 identical strands, but in opposite directions. So in other words, the strand on the right, this side right here, is the same as the one on the left. It's just upside down. Now imagine this for a minute. If you peeled this apart like a zipper, if you opened it up, if you cut this right here, separated the 2 halves, and then built 2 new halves on each side, you'd have 2 identical molecules, 2 copies of the original. Let me show you what I mean. Take this apart and take this apart. So now I've got, almost done. Now I've got this here. I'm sorry, this should have been an A, right? A, G. Now, by the way, the order, it's going to be different. For different genes, the order of the letters is the gene. So this goes C, T. And now the other half, if I split these apart, the other half will look like this. And it'll have the bases coming off, but it'll be T, C, G, A. Now, let's say I had a bunch of these individual units, which are called nucleotides. Let's say I had a bunch of nucleotides floating around in the nucleus. A bunch of those pieces. They would actually be attracted to each side of this thing, and they would start to match up like this. And it would actually build a whole new side on this thing. One by one. So picture these pieces right here. These pieces. Picture them floating over here to this to this side here, and they're going to build two new identical structures.
What does that say, nucleotide or nucleotide?
Nucleotide, sorry, let me clarify that. Nucleotide. A nucleotide is a phosphate, sugar, base. Just one unit of those three molecules. So these are actually floating around in your nucleus as we speak. And if you split a DNA molecule apart, they will naturally be attracted to each broken side and build a new side on there automatically. Okay? So that's what happens. Now the same thing happens over here. You get A, whoops, G, C, T. And so you end up, out of this whole process, you get a whole new molecule, actually two new molecules, that are both exact copies of the original.
Are we going to have to draw these?
I don't think I'll make you draw it. I would probably draw a picture and say, what's this part, what's this part.
Oh, like A, C.
Yeah. Now, that's just an overview of this section in the book, which is what's the basic structure of DNA and how does it replicate itself. I will show you, I've got a nice little site that I'm going to put on Moodle. Right now it's just on my e-board from last year. But I've got some things on here that you can play around with. Let me just show you what I mean by this, this replication process. How long has it been? It's been about 10 minutes. It's been about 10 minutes. Okay, so let's see, DNA. I'll put a link to this up there because you can try this out. So what we're going to do here is we're going to try to replicate a DNA molecule. And this takes you through kind of a description of it. By the way, the helix idea is that those two strands are twisted, as you can see in the models, into like a spiral staircase. It's like a spiral staircase. Do you ever see a spiral staircase? Who's seen a spiral staircase?
They're really dangerous.
You know they go down.
They are dangerous. I have a cousin who actually fell down one time. He had food poisoning and he like passed out at the top. And he got hurt pretty bad.
I was in a
He fell
haunted
straight
house,
down the thing.
And one of the things in the haunted house was a spiral staircase. And it was a scary part.
Wait, you fell down what?
A spiral staircase.
You know, in most.
All right, watch this. This game is fun. Watch this. Are you ready? Here we go. We're going to start to replicate. This thing comes up. The two sides of the DNA come up and you have to match them up. So C goes with G. C goes with G. G goes with C. G goes with C.
You're a loser.
C goes with G.
You're losing, bro.
G goes with C.
No,
Oh,
no.
you're done.
No.
With C.
No.
No.
C doesn't go with C.
All right, my T. Hey, hang on. Oh,
Oh.
You're gonna do one side.
One.
And you're the bias. You froze it.
It's laughing.
no. All right, anyway, you could try that on your own. You can try that on your own. All right, last thing before I end, folks. Just so you know where we're going with this, let's say this is a strand of DNA. Look at this. Listen up. This is it. And then we're going to introduce number 24. Here's where we're going in the next section. What's the whole purpose of this? Those sequence of letters, I've been putting it in one order, right? It's not always in the same order. Excuse me. Folks, it's not always in the same order, and it stands for something. And here's the deal. Every 3 letters stands for one of 20 amino acids that are used to build all the proteins in your body. Every 3 letters stands for one of 20 amino acids that are used to build the proteins in your body. And in the next section, we talk about how the cell translates that code into proteins. And then the proteins control things like the growth of your cells, color of your eyes, color of your hair, how tall you're going to be, whether you're going to be a chimpanzee or a person and all that kind of stuff. Okay? So this code contains the information that's used to build your body, essentially. All right? And your brain and whatever else. And so this code has to be copied exactly. All right? And I'm going to get you started on the next